Hi, this is Sureka. Today we're going to see a lecture on transcription, which is a synthesis of RNA. So, when you come to the definition of transcription, transcription is defined as a synthesis of mRNA from the DNA. So, the information stored in the double-stranded DNA will be converted into the proteins during the cell multiplication or during the cell synthesis. So, during the conversion of the DNA into the proteins, the information stored in the DNA will not be directly transferred into a protein. It will be first converted into mRNA, then it will be converted into proteins. So the conversion of DNA to mRNA is referred to as transcription. So the information stored in the double-stranded DNA will be converted into a single-stranded mRNA. In the process of transcription, then the mRNA will again be translated into proteins. So when you come to the enzymes involved in the process of transcription in prokaryotes, there is a special enzyme called DNA dependent RNA polymerase, otherwise called as RNA polymerase. So from the name itself we infer that the RNA polymerase, it requires DNA template to synthesize mRNA. So DNA template is required. It uses one of the strands of the DNA as a template stand to copy down the sequence to form a mRNA. So when you see the structure of the RNA polymerase, it has got four subunits, that is two alpha subunits, one beta and one beta subunits. They all together they are referred to as a core enzyme, and it has also got a detachable subunit, that is a sigma factor. This sigma factor has a potential a role in RNA polymerase. It does a recognition of the promoter region in the DNA template. So it helps the RNA polymerase to recognize the promoter region in the DNA template. And once the sigma factor attached to the core enzyme, then they are collectively called as holo enzyme. So when you come to the stages of transcription, it has got three important stages, that is initiation, elongation and termination. In the stage of initiation, the RNA polymerase with the sigma factor binds to the promoter region in the template. So the sigma factor in the RNA polymerase helps in the recognition of the promoter sequence where the RNA polymerase bind to it. And the promoter sequences was found to be varying in prokaryotes and the eukaryotes. So when you come to the sequences of promoter sequences present in the prokaryotes, this is of two different region. One is Prinov box or -10 region, the other one is -35 region. So -10 region consists of the nucleotide sequence data, and it has got a base pair of 10 base pair upstream to the start point. And at -35 region, it has got a nucleotide sequence of TTGACA, and it sequences 35 base pair upstream to the start point. So the promoter sequences are responsible for directing your RNA polymerase to initiate the transcription at the particular start point. And this shows you the role of the core enzyme, that is your four subunit. See here you have got a promoter region, that is -10 consensus sequence. This is -35 consensus sequence. Your RNA polymerase with the core enzyme, RNA polymerase with the sigma factor tries to bind to the -35 sequence. It first recognizes the -35 sequence. Once 35 sequence gets recognized, it just helps the RNA polymerase to move forward to recognize the -10 region. It just indicates or signals your RNA polymerase that there is a start point after 10 base pairs. So your RNA polymerase binds here and start unwinding and start the process of initiation. So your core enzyme helps in the formation of phosphodiester bond between the ribonucleotide, complementary to the DNA template stand. Thereby your initiation gets started. So when you come to the stages of elongation, it just proceeds after the formation of first phosphodiester bond. So your RNA polymerase core enzyme starts adding the ribonucleotides, thereby a small transcript mRNA new transcript will be synthesized. So when 10 phosphodiester bonds formed, your sigma factor get dissociated from this core enzyme, and your RNA polymerase start adding up or elongating the mRNA transcript just by adding the ribonucleotides. And your process of elongation of the RNA continues until the termination signal is reached. So this is how it happens. See here the sigma factor with the 4 subunit. It just binds to the promoter sequences where it gets recognized, and once it gets recognizing the promoter sequence, it just starts moving on the DNA template, and it starts reading the start site where it can unwind and start the initiation of transcription. After the formation of the small DNA, I mean mRNA transcript, here a sigma factor will get dissociated, and even after the dissociation of sigma factor, the mRNA will be elongated just by your core enzyme. It just adds the ribonucleotide and elongate the chain of mRNA until the reach of the termination codon. So in the process of termination, in prokaryote, it involves two important mechanisms, that is pro dependent and rho independent. In rho dependent termination, the involvement of rho protein, which is a ATP dependent helicase enzyme, which is a protein that can bind to the mRNA transcript. The mRNA has got one specific site for the binding of rho protein. So this mRNA, the rho protein gets bind and just climb up and reaches the state where the RNA and DNA were forming the hybrid structure. So this goes and reaches this point, and it just unwinds this because it has got a helicase activity. So it just unzip or unwind it, leading to the termination or leading to the dissociation of the mRNA transcript. When the mRNA gets terminated, the RNA polymerase displaces from the DNA template. So this is how the rho dependent termination happens. So when you come to the rho independent termination, where the formation of hairpin loop occurs, this occurs due to the presence of the palindromic sequences in the newly synthesized RNA, which removes the RNA polymerase from the DNA template, resulting in the formation of the transcript. This hairpin loop structure usually followed by the 4 uracil residues essential for the termination. The RNA transcript ends within or just after that. See here, after the uracil formation, there is a GC rich region. This GC rich region, they form a hairpin loop structure in the newly synthesized mRNA. Once the newly synthesized mRNA is formed, hairpin loop structure is formed in the newly synthesized mRNA due to the presence of the GC rich region. There is a halt in the movement of the RNA polymerase. This results in the breakdown of the bond between the A and U, leads to the termination or the release of the mRNA transcript. So when you come to the inhibitors of transcription, the synthesis of RNA is inhibited by certain antibiotics and toxins, say such as actinomycin G or dactinomycin. This is synthesized by one of the species called streptomyces. They can bind to the DNA template and blocks the movement of the RNA polymerase, and this is very first antibiotic used for the treatment of tumors. And other inhibitor is rifampicin. It is an antibiotic used against the disease tuberculosis and leprosy, and they find to bind to the beta subunit of the prokaryotic RNA polymerase and inhibits its activity. Here the picture showing the rifampicin 300 milligram capsules. So with this I'm ending up the lecture, and thank you for the opportunity given. Thank you so much.